So now we need to focus on algal biorefinery. So my first project is like what I'm gonna discuss is today is basically a recovery of microsporin like amino acids. And second, I'm gonna talk about a process where we can change the process and parameters to recover maximum of the products. And third, I'm gonna introduce with something which which is looks sustainable in near future. And fourth one, I have added one more thing which is related because I know most of the students from JNTU are from engineering background. So for them also, I added one more topic. So there are four topics in this presentation. So now, if we talk about the sustainability, so there are four things we can do. First, we can increase the cell density because increasing cell biomass is one of the constraints. So if we increase the cell density, like you are producing a lot of dry wet dry biomass from algal cultivation, then you can overcome lot of parameters. Like if productivity is less, you can satisfy that demand. If another product, you can use that biomass for multiple products. And second, you can say like increasing the intracellular content of desirable products. For example, if you need any pharmaceutical active product, you increase the amount of that product. Or else if you enhance the biodiesel content in it. And third is can be both, which I will say is the best way. Like you increase the biomass and at the same time you increase the desirable content, that also. And fourth is, I will say, to find a method which is very helpful for extraction of these products. And these methods should be like eco-friendly in terms of what solvents we're going to use it. Plus, it can be easily performed by at the industrial scale, because what in lab we were doing, it's not that much reliable that we can produce at a huge scale in industries, because we are using a lot of solvent, chloroform, methanol, hexane. So these are sometimes, if we handle chloroform in bulk, that you need that infrastructure, or else uh, that may be dangerous. So we need to work on the methodology too. So there is, I will introduce to one methodology which I worked recently. And so considering all this point, I will say biorefinery is one option where we can recover the products efficiently. So this is the first work I'm going to present, where I'm going to focus on production of MAs like microsporin like amino acids from microalgae along with the biodiesel. So MAs are basically, they are kind of UV protectants, like we use the sunscreens to protect from sun or suntan. So it's a similar kind of product which we can recover from microalgae, and they are actually water soluble. And when we consider the lipids, it's soluble in organic material, and MAs are soluble in aqueous form, like basically water. So this is the one of the thing with which we can separate these two compounds from a single biomass. So how I proceed with it, I took the algal biomass. Here we took the lingua, and then we exposed it to UV light, which is basically UVA and B. And then we extracted with the 100% methanol. And after the methanol, we evaporate the methanol and take the residue, and that we dissolve in the water. So once you dissolve in the water, there will be all MAs dissolved in the water, and the all lipid components will be on the upside. And once we'll add the chloroform, it will dissolve in the chloroform. So basically, we created two phases. One is aqueous form and one is chloroform form. So aqueous form is having the MAs, and the chloroform phase having all the lipids. So that way we separate both of them, and then once the aqueous phase, we will evaporate the rest of the water and we will come up with the MAs and take the chloroform, and from that we can extract the lipids. So this is the hypothesis that we proposed for it. This is already published work, so you can read in detail also, but I'm going to explain it here too. So basically, we studied two wavelengths. One is UVA and UVB. Why we choose the UVA and UVB? Because these two are the wavelengths which comes to the Earth atmosphere. Because UVC is absorbed mostly by the ozone. So UVC is not coming on the atmosphere. So if we are proposing the, then it will not be that much helpful. So I calculated the energy, how much energy they have, what are the conditions, and UV type, and then there is a like time of exposure. I studied for two times. One is 12 hours, and another one was 72 hours to see what is the impact of uh, these timings or exposure dose on these algal cells. So when we come up to the production of it, so we can say easily UVB shown the highest production of MAAs irrespective of the time, because 12 hours and 72 hours both are giving a higher UVB productivity rate. UV has a slightly less of UV MA's production because UVA have a lesser energy than UVB. So we can say more the energy of UVB intense radiation, then MA's productivity is high. Once we extracted these MAs and we quantified on like a dry weight basis, we go further for the absorbance, like really we what compounds we extracted, they are MAs or not. So actually they have the maximum absorbance close to 330 nanometer. So UV, when we took the content from the UVB 72 hours and 12 hours, they have the maximum peak. So that shows there is MAs and which is showing the absorbance at that particular wavelength. So this was the preliminary test for our MAs. Then I go for the FTR analysis and try to find all the functional groups what are present in them. So we come up with the almost of the functional group which is related to the MAAs, and we got this profile. And all these functional groups are mostly associated with MAs compound microsporin like amino acids. And later, once we quantify it, like yes, we extracted the MAs, then we started studying the efficacy of these compounds. Like these are like they have a biological activity or not. So we go for the antioxidant activity. So for antioxidant activity, we took the Trilox equivalent and then we quantify. It's a very easy process. Like sometimes there is available kits also. Sometimes we need to prepare the reagents. So when we see the efficacy, so the UVB radiation has shown the maximum antioxidant activity. This is also because of they have a more of MA's productivity. But at the same time, when we see the biomass concentration of different UV radiation, at UVA, we got the higher biomass, but at the UVB, our biomass productivity decreases, which we predicted that might be because of the UV radiation, UVB radiations created a stress on algal cells, and that results in decrement of the biomass. And after this biomass, we come to the main, the one of the most important topic, which is the lipid. So here, when we took the total lipid, we quantified the total lipids, and yes, UVB still show the lipid productivity, but UVA was having higher lipid productivity, which is close to 30 mg like per mg of dry weight, which is actually it is 30%, you can say. So that is quite good lipid productivity when we consider in a biorefinery platform. And after that, we did a fame recovery, but the fame recovery was UVB and UVA were very close. So we can say like the good kind of lipids, which is for the biodiesel, is good there. It is in UVB still it's there. So these strategies work well to recover the MAAs and the lipids together. And after that, what I did a like a lipid profiling, And lipid profiling was really interesting because we need most of the saturated fatty acids when we talk about the biodiesel. And our UVB actually shown maximum of the C16, like we can see in the figure also, C16, which is like a good kind of biodiesel mm, property it have. So that concentration was higher in UVB. So however, the lipid productivity was higher in UVA, but the good kind of biodiesel productivity was higher with UVB. So UVB is good in both way. If we talk about the like a pharmaceutical active product like UV, like MAs, and along with the biodiesel productivity, having a saturated biodiesel content. And after that, I did a mass balance. This is based on the COD mass balance, where we convert everything towards like in COD. It is carbohydrate, MA, cellular proteins, phycobility proteins, and everything. We convert it to a COD thing, and then we see how our mass is moving between one part to another. So here, if you see this red one is MAAs, like one is like a dark red carbohydrate and one is red MAAs. There was very slightly variation. That is an important component because they're pharmaceutically active compounds and their minute concentrations also plays a major role. But when we talk about the saturated lipids, they are the major content of UVB radiations. And slightly the cellular proteins was also high in UVB but lower in UVA. And one thing we saw, sorry, is the phycobility proteins. If you see the blue one, phycobility proteins actually what they do, they're kind of protein which are like accessory photosynthetic pigments in cyanobacteria, which is which acts like antenna pigments for the solar energy recovery and to proceed the photosynthesis process. And that was also like significantly higher in most of the thing, but our control, which is photosynthetic active radiation, the last one was having the maximum phycobility proteins. So now the second topic what I'm going to talk about uh, is the process, like using how with the process we can recover maximum of the products. So I'm going to talk about the electrofermentation. There are various solvent strategies are there. We using different kind of solvents, using different kind of processes to extract it, like success operation, or simply like extracting everything in a solvent mixture. And this is a slightly different. It's electroselective fermentation. Where here what we did, we ferment the algal biomass in a microbial fuel cell where they can have a proper conditions for fermentation plus biohydrogenations and lipid breakdown too. I'm going to go in detail of it. So first what I will say, see this process. This is like a H cell microbial fuel cell. This green one is anode and this white one is cathode. And this anode have anode respiring bacteria. And in the same one, we put the algal biomass. So this circle represents the whole algal biomass and it have all the carbohydrates, proteins, lipids, neutralipids, unsaturated, everything is there. So once we go for the fermentation, the cell wall breaks down and everything comes out. But everything comes out slightly in a different form. Like there is a VFAs, there is a saturated lipids, there are some glucose molecules too. So what we are projecting here, if we if in this anodic chamber there is a protein and carbohydrate and we do the electrofermentation, it will result in VFA and hydrogen. And what will happen, the unsaturated fatty acid which is present in the algal cells will react with the hydrogen here, which is produced by the fermentation. And this hydrogen will result in direct biohydrogenations. What will happen during the direct biohydrogenation? All the unsaturated lipids will convert to the saturated lipid. So what all the lipids present there, it's converted to our desirable lipids, which we can go for biodiesel. And there is a one more phenomenon happen. The unsaturated lipids, they will go for beta oxidation. Beta oxidation is simply like a breakdown kind of things will happen, and the unsaturated will convert again to the saturated lipids, and there is a formation of some VFAs hydrogen too, and this hydrogen will again circle to the direct biohydrogenation. So this was the hypothesis initially we thought of it, and then we started working on it. So this is the first data, the current density and time. We maintain a constant potential of 200 millivolts. So what will happen initially? The system has a lot of fluctuation. So from here till like 15 days, we can say system was not at all stable. So we ignored the data from this time. We took the data after the 20th days, from 20 to 25, when the system was very constant in its performance. So we did initially the COD, so total COD and soluble COD. So this top, this one is like it's like total, and this the bottom is like soluble. So FB is feeding biomass, and this is selective fermentation, and this is electrofermentation. So once we see the electrofermentation, their biomass is the COD is actually decreasing. It means the content have degraded and it's converted to some other form. And when we talk about the soluble COD, it means its soluble COD is getting high. It means that things are fermenting. And this is a comparison with the feeding biomass, which was initial, and the selective fermentation without any electrode setup, and with the electrode setup. So with this figure, we can say when you put the electrode setups in a fermentation tank or fermentation vessel, there is a higher degradation of total COD, and that results in increment in dissolved COD in the system. And then what's going to happen to that COD later is like this. So with electrofermentation, what we see the carbohydrate and protein, both are there. In selective fermentation also, carbohydrate and proteins are there. So basically, the carbohydrate and protein both are degrading in electrofermentation, and that degradation will result in the hydrogen and further degradation of further biohydrogenation of lipid. There, like any student can ask, like protein is degrading, carbohydrate is degrading, but why not lipid is degrading at the faster rate as carbohydrate and protein? The answer will be like the lipid degradation takes time, but protein and carbohydrates degrade faster than the lipids. And when we operate the reactor, we have to design the hydraulic retention time at a level so that most of the organisms which are present there are protein degraders or carbohydrate degraders, and we need to avoid the lipid degraders, and that most of the time come in the later phase. It's the same like human metabolism also, like protein, like you get an instant source from energy from carbohydrates, but for proteins it will take time to get any energy or degrade further. So this is also a COD mass balance. Like this is like 100% here, I wrote it, and this is with respect to the all mg respective amount of CODs at every stage of feeding biomass, selective fermentation, and electrofermentation. So if you can say there is a there is a degradation of the carbohydrates and proteins, and everything is moving towards the lipids profile. And after that, once we analyzed that yes, lipid is producing carbohydrate proteins are degrading by hydrogenation. Like at the end of our experiment, we did this thing, TEM analysis. Like we took the whole algal salts and we like did the TEM, and here you can say the cell wall is intact, but slowly, like once we start degrading them, their cell walls start loosening up. So it means the cell wall is degrading at a faster rate, and that is a result in accessibility of more of the desirable compounds. And then we did the community analysis. So we have this is cyanobacterial and plus DNA we got because we have took the microalgae. Here we say the more of carbohydrate fermenters, which was really high, and protein fermenters are high too. But there are one section which is called lipid fermenters. They are really low. So it means we are successfully operated the HRT to degrade the protein and the carbohydrate so that we can have the maximum lipid productivity. So actually, if we modify the processes like this at higher scale, then with a slightly like not so harmful solvents, we can get a good lipid productivity by this process. And if this will be get integrated to a biorefinery, it's easy to recover other products. With this project, we also working with the recovery of pigments. And like we got some good data, but still we didn't publish it till now because some more testing is left. But yes, these kind of things will be easily integrated for the recovery of the pigments too. And viable pigments, it's not like these pigments when we extracted and none of their use. They are active, they have a bioactivity too. And after this sorry. Um, After this, I worked on one more microbe, which is called Cyclotella, which is a diatom, which it can be grow in any kind of wastewater. But when we start growing a pure culture, they are very hard. Like they don't grow very well in pure culture, but in most of any kind of wastewater samples, you can get these kind of organisms very easily. Cyclotella, which is like when I worked in Hyderabad, like the wastewater what we got, it has a lot of cyclotella and other diatoms. So, but this study we focused on like a very basic research. So not like a basic in the terms like fundamental research. So we need a pure culture for that. So we took the cyclotella for that. And why we took the cyclotella, like there is a what we think a theory behind it. Like the present day, what all the diesel we are using, we say it's from petrochemical rocks and like I don't know the years back, it's get buried and like these diatoms result in the formation of these reservoir like uh, oil wells. So the years back, if those organisms can produce that diesel, so what if we use the same organisms and start producing the diesel right now? So I took this cyclotella because they have the fossil remains from the past also. So I targeted that. And what we did initially, because these cyclotella and diatoms they grow with the in presence of silicon. So this like initial things we did, we monitored the bio cell density and we see how the silicon is depleting. So it was like a control kind of system, like to see like they're growing in presence of silicon or not. So yes, they grow, and this is the reactor what I used for the cultivation. It was like a 15 liters reactor to grow them. uh, and whenever you're gonna grow diatoms, just follow the proper protocols, because if one mistake, they might not grow. They will grow well in the wastewater without any issue. But when we start growing in pure culture, it's hard. So with this, I grow them for like 8 days, and we took the lipid productivity. So they have quite good lipid productivity, like 50%, which is really good. So with 50% of lipid productivity in very short span of time, just 5 days. So we took, we extracted and we profiled it, and like at 8 days they reached their maximum productivity. And after the total lipid productivity, we see the saturation index, and actually they have a very good saturation index, maximum of 80%. Like even all, like after 5 days, all have a 80%. It means what the what all the lipids we extracted, 80% of it is useful for us for the productivity of these biodiesel. So it was a very positive sign, because when we work with the mixed culture, they have the lipid saturation index dropped to very low, sometimes like 20%, like 50% to 20%. So in between. But if it's 80%, it means what all the diesel we extracted, we can use them for the biodiesel. And one more interesting thing came up when we did the lipid profiling. Because if you do the lipid profiling of today's diesel, like what kind of uh, carbon chains are present in that, they mostly contain C16 and C14. And when we did the lipid profiling of these diatoms, they are mostly of C16 and C14. It means their composition is very much similar to the diesel what we are using. So this was like interesting for us, and we're still working on it. But yeah, let's see when this lockdown will finish. I'll start again. And after this, um, what we did, we took the fructose too. Like once you extract the lipid, you come up with some of the residual biomass. But in case of diatoms, it have a frostule. Frostule is like a cytoskeleton of diatoms. I will show you in the next picture. So when we analyze the FTIR of all these diatom frustules, so they are made up of silica, obviously, and all those functional groups pop up. And then I did a elemental analysis. Here the silicon was maximum, and I slightly get oxygen and carbon because maybe there was no proper washing. That's why some organic content still stuck to those frustules, but the maximum was of silicon. So once we are able to extract these silicons, frustules, they have a lot of application. They have biomedical application too. So this is the work actually one of I did in a with MIT, like for a short research stay, but still we are doing it further. So this is the picture what I'm talking about. This is the towards the left side is the whole diatom frustules with all the biomass intact to it. But once we extract the biodiesel and we took the frustules, all biomass are like 80% of the biomass is gone. Still there are stuck something, and these are like at a scale of nanoscale, so this can be used as a drug delivery factors, or like they have a lot of biomedical applications. So this is also a biorefinery. You are getting diesel, you are getting the algal skeleton, which is at the nanoscale level, and you can use that in a drug delivery platform. So this is the one of the thing. And then I added this point because I consider like a lot of students at JN2, they are from the engineering background. So this is a typically of for the engineering students, where we use the food waste in a leached bed reactor for its degradation. So basically it have a, we try to produce bioenergy, and we try to decrease the solid content. Like in food waste, the whole solid content, how we can decrease it to or minimize it. So this is the right hand side is the reactor, how it look like. At the top there is a bucket and there is a sprinkler, and at the bottom it's all at the leachate. So this is the reactor diagram. This is the leachate bed reactor, because at the bottom it's a leachate. At the top there is a bucket, and there is a sprinkler over here. We put the food waste here, the solid food waste, without any mix, like without any grinding or anything, just as is food waste, just chopped in like some small pieces and just fill up here in the basket. And there's a leachate which circulates the leachate from the top. It just sprinkle over here, the leachate degraded, and again it come at the bottom. So at the top, it's a solid food waste, and the bottom is the leachate bed, which have all the microbial population cocktail, and it the microbial degradation actually takes at the both the places. But the major objective with it is to decrease the solid content. So this is the simple like the the biochemical process is simple as normal fermentation. So this is the way we feed the reactor. So the first three cycles we go for the defined food waste, and then the last three cycles we go with the real food waste. So the TSS was TSS, VSS, all those like wastewater treatment uh, protocols we follow to quantify the soluble COD, total COD, TSS, VSS. So I give the parameters here. So how we feed the reactor. So firstly, we take the food waste from we took it from the cafeteria. We removed all the solid parts which cannot be degraded, and then we just cut in the form of a cube of 1 centimeter cube, and then feed to the reactors like 1 kg, 1.2 kg every time, and we maintain inoculum ratio of 4%. Like 4% will be our sludge, and rest will be like food waste. And then we measured the COD, BFA, TS, VS. This is the basic analysis what we do. Along with that, we do the biogas monitoring. And we end up with the solid residue, and one will be the leachate, which gonna be very high in volatile fatty acids. Here we are able to achieve high VFAs because our food waste was not at all diluted. It was as is. So um, its loading rate was very high. So after that, then when we started analyzing the VFA content, so run 1, 2, and 3 are basically with the defined food waste. But in the run 4, that was a real food waste, we are able to get a higher concentration of acetate and butyrate, which was most of the time you will get these two VFA. But once we talk about the biorefinery, we think how we can enhance the productivity of these VFAs so that we can extract them. So we successfully able to do that to enhance um, the concentration of these VFAs in the leachate. So acetate and butyrate was highest. And then after that, we did like a cost analysis, like how much energy we put in that and how much of energy we recovered from it. So basically, it was based on the small LBR reactor, and which we extrapolated in terms of tons, because we just recently got a project from an industry where we are working on minimization of this food waste to a higher extent, and how much energy and income and outgo it should be there. So at a smaller scale, we feel it was a cost effective, though it was not that much, because if we here the cost benefit like is $1.76 per tons of VSS added, which was actually negligible. But the thing from here we are working on it and further, and how we can decrease the input cost and how we can increase the output cost, that are still we are working. So this preliminary data is already published, so if anyone wants, they can go for it. And along with it, we studied the pH, and we studied the microbial community, what's present over there. So I will directly go to one here, this one. This is at the pH 6, and there is a one thing by feed your Cyanobacterium. Yeah, here. And actually, these are the class of microbes which degrade fibers, because the food waste, what we used it as is, was not at all cooked. It was like most of the vegetables, peels, and we put as is. And there, these class of bacteria enrich a lot, and which are the fermenters of fibers. So thinking about it in a large scale, when we use this technology, this uh, class of bacterium going to play a major role. And we are also thinking like bifidobacterium will also be helpful when we talk about lignocellulosic waste, like all the waste from agro-industrial platforms, because they have a lot of fiber-based waste, and they can be a key player for the degradation of these fibers and like solid reduction. So this was the major finding with these microbes. And obviously then we studied the various pH like 6, 7, and 8. And if you see, we are able to get a bute rate of 16 grams of COD, which was really high. This if our VFA concentration is high, it's easy for us to extract them from the like media or from the waste, and we can make a good biorefinery platform from it. So this thing, I just went back to explain that slide. So major thing is like with this technology, yes, it's feasible to degrade the waste like this, which have a lot of fibers or agro-industrial waste, and with a minimal mixing, you say, or grinding, because we can use the as is. And if bifidobacterium is the key role player in the degradation. So with this, I would like to conclude my studies. So first one with the UV protectants. So yes, it is feasible to produce MAAs like mycosporine like amino acids along with the lipids. And yes, MAAs have UV protectants activities, and which can have a further application in biomedical industry. And second, with electrofermentation can provide a good uh, process for a degradation of algal lipids, which helps in production of saturated fatty acids, along with avoiding like harmful chemical solvents, basically for this extraction, and end up with a good kind of biofuel. And third one is from the diatoms. Yes, we can take the advantage of the natural selection, which before already it happens for the diesel, what we are using it. Is nowadays we also work on diatoms, which are very uh, potent in producing the lipids and the further saturated lipids for the biodiesel. So yes, at large scale industrial setup, diatoms can be very promising for biodiesel industry. And third one is from my LBR reactor. So the LBR reactor is really good in degrading, in minimizing the solid waste. And when we talk about the pH and all the VFAs production, acetate and butyrate was the maximum there. And we can enhance the fiber degrading bacteria, which is bifidobacter. And this class of bacteria is also helpful in future research when we talk about the lignocellulosic waste or any kind of fiber degradation. So with this, I will finish it. And at the end, I wish to give my email address. If you have any questions, you can write to me. And this is my Google Scholar. You can get most of the paper, what all the papers I presented, most of them are published, except the diatom one. So you can get all the details from there. And with this, I wish to conclude it and open for any questions. If anyone has
Can you
any
read the questions?
Yeah.
Sushma?
Yes, ma'am. Yes, ma'am.
Please read. Start.
Yes, ma'am. Ma'am, there is a question. any green technology instead of solvents for extraction and recovery of lipids or byproducts from algal biomass?
So please tell
Okay, now I
the
guess
participant name, Sushma. Please tell the participant
Uh,
name, then read the question.
okay.
yeah, I can read the question here. It's from ISTC and ISTJ. Yeah. So the question is, any green technology instead of solvent for the extraction and recovery of lipids and other byproducts from algal biomass? Yes, there are. I have a paper, you can see it. It's with the CO2, like what we do. It's a critical, like you use a critical temperature and using CO2. So what you take a liquid CO2 and then you pass it to the algal cells, which is like a critical biomass extraction. We published it where somewhere in 2016, where you can use simply as a CO2 as a solvent for extraction of lipids. But for that, you need an instrumentation so that it can hold that temperature and pressure because you have to minimize the temperature and you have to increase the pressure. So that is already there. So with this technology, you can use this extraction of lipids. Along with the lipid, you can recover even the proteins because that is very feasible technology. But the thing is that that instrument is costly. Yes, so this is my answer.
Okay,
And the
ma'am.
second
There is a
question is participate if any question. Okay. And next question is, ma'am, extraction of lipid process is either done by phase separation or by any other method. Extraction of the lipids from the process is either you need some extracting reagent in order to extract lipids. Yes, you need like extracting reagents most of the time, except if there is supercritical fluid extraction. With that, you don't need like these much of the solvents, but you need it. But the thing is that if you break down the cell wall or you do some pre-processing, then you can go with the slightly eco-friendly solvents. For example, if we are using chloroform methanol, which is considered as a gold standard for lipid extraction, but what about if we use something like isopropyl alcohol, or we use something like hexane, normally we use for the neutral lipids. So yes, but the thing is that you have to first work on the pre-treatment, and if your pre-treatment is really good, it's easy for you to get to extract the maximum product recovery. And for example, if you take a chloroform and methanol, like 2:1 ratio, and you're extracting the things, and if your cell wall is not broken properly, then there is a possibility your all lipids will not come out properly. So yes, you have to do the things beforehand. And then there is a question again. Okay. There is a stage during the process where biomass growth and productivity decrease. I think you asked this question, like I will read the questions for everyone. Ma'am, is there any stage during the process where the biomass growth and productivity decrease? So if we talk about the UV protectants, yes. If with the UVB, the biomass productivity decrease because UVB is a high energy radiations, and in high energy radiations, um, the cell has to survive. So most of its energy goes to its survival and it's making MAAs. So their overall biomass productivity decreases. So yes, it is there, and that is there. And with diatoms, I didn't get any decrement with because I was growing it in a pure culture, so it was growing well. And then there is a one more question. No, there is no question. There's a suggestions. If any other question, you can ask. No questions? If someone
in
is
the
shy,
chat
they can
section, there
type
are
it here. Like
Yeah,
I can see up to this 9:16. Hey, Nikhil, good to see you.
yeah, good to see you. Nice presentation,
Yeah.
but I missed most of the slides. Have been some of them, but
You were late or what? You will have to
I
be
should
on
thank
time.
Professor Himabindo Madam and Venkat Mohan sir. It's a kind of alumni meet.
No problem. It's okay. You can write me anytime.
Yeah, sure. I just have a query. What about any syngas kind of fermentations using algal systems? Any possibility?
Yeah, actually, if we prolong it above 24 hours, then there is a syngas kind of. But we want to restrict it for hydrogen so that we can have a maximum of bihydrogenations. That's why we are making it to 24 hours, and we are maintaining a COD of 8 grams per liter. So with that, we are getting mostly of hydrogen. But if we leave the reactor for a long time, they will start producing initially the mixture of hydrogen, methane, and then completely of methane. But if methane is producing, then our most of the glucose and other things is degrading to other things. Plus, our lipids also start degrading. So at the same time we have to preserve the lipids.
Okay,
It's there.
great.
Too. Yeah.
Okay, nice.
Any other question?
Yeah, best wishes
No?
for the future.
Thank you. I need a lot of. Amazing.
So
Thank you.
I think that's all. I think no more questions. I think so. Lakshmi, you were able to degrade that one, the carbohydrates and proteins
Yeah.
to keep the lipids.
Yeah.
So can until like if you just degrade the carbohydrates and proteins are also useful materials, no? Like
Yeah.
so
There.
can we take the carbohydrates out and then leaving the proteins and then lipids? That can you separate them?
Actually, we are working on it. Like for
Uh-huh.
example, once we remove the lipids, then
Uh,
now we are working how we can decrease the HRT, and we can also recover phycobiliproteins.
yeah.
Actually, we are able to recover chlorophyll A and B with this experiment also, but that was not that helpful. Once we have something good protein like phycocyanin or phytoerythrin, when we work with the cyanobacteria, so it is possible. But again, we have to optimize the HRT as per as.
Hmm.
So it is possible. I mean,
Yeah.
that's good too.
Okay. So then if there are no more questions, then so thank you, Rashmi. You have covered in situ like this how to produce the saturated lipids, the methods, and then
Thank
how
you.
to eliminate the other impurities in the lipids to go for biodiesel. That carbohydrates and proteins removal will help the good amount of not the efficiency of the biodiesel will be improved if you remove the contaminants like proteins. So I've explained all these methods well, and then I hope the participants have gained some knowledge, the students particularly, so they can choose some of the fields in this line. I hope.
I hope. I
Yeah.
hope this is helpful for all the students and they
Yes.
get some lead to work on
Yeah.
something.
So in that is the requirement nowadays for look for waste as a resource, and so we have to proceed. So thank you very much.
Thank
Uh,
you, ma'am.
yeah,
Thanks for
yeah,
everyone.
yeah,
And thank you, ma'am, for considering. And thank you to Venkat Mohan sir
yeah,
too.
yeah.
I can't
So,
see him, but yes, I'll appreciate it.
yeah. Okay, so today we are going to end this session, and tomorrow we'll have 11 o'clock onwards. We have three good lectures. Dr. A.G. Rao Garu from IICT and Dr. N. Mitra from Cambi, India. So, and another Gopal Krishnan from Norway. So three presentations we have from 11 o'clock onwards tomorrow. So we will meet tomorrow at 11 o'clock. Okay. Thank you,
Sure.
one and
I'll
all.
try to be there.
Thank you, Rashmi.
Thank you, ma'am.
Thank you.
Thank you. Take care,
See you.
everyone. Bye
Yeah.
bye.
Bye.